Hi, everybody. I'm in a meeting, but I want to make sure you keep up with all the other classes. So let's go over the starter for today. We're working with the same gene as last time in hibiscus flower. So we know that red, the color of the petals, is dominant to white. So we're going to use capital R for red, that form of the allele, and lowercase r to represent the recessive form of the allele. What I was hoping you would do is use vocabulary from last class, your notes, or that practice page I gave you guys with the dogs on the back to choose the appropriate vocabulary that describes big R, little r, so you can just get a little more familiar with the way I'm going to describe them in class and on future assignments. So for big R, we would describe that as either homozygous dominant or purebred red. And we do have to include the word dominant or red in this because if we just say it's homozygous, that just means that the two alleles are the same, but it doesn't specify which one we're talking about. However, when we say homozygous dominant, it means that they're two capital letters. For uppercase R, lowercase r, we would describe this as either heterozygous or hybrid. And we don't need to say that this is dominant. We also don't need to say this is red. It's assumed to be true. Since there is the dominant allele present, it means that these flowers will always express that. It's not a blending of red and white. It's not going to look pink. We're talking about a situation where it's either dominant or recessive. And in this case, the dominant is expressed, so they'll always look red. With lowercase r, this can be either homozygous recessive or purebred white. We could also just say it's white. It's a white flower, and know that both alleles must be the recessive form, lowercase r. So it's homozygous recessive, purebred white, or just white. So what would they look like? What I'm really asking you here is the phenotype or the physical appearance. What do you see? And in this case, it's only ever going to be red or white. So for genotype big R, homozygous dominant, that's going to have phenotype or physical appearance red. And the same is true for your heterozygous plants. If it has genotype big R, little r, the phenotype, the physical appearance, what you see will also be red. The phenotype for little r is, of course, going to be white because that dominant is not present. Therefore, it's going to appear as the recessive form or the white flower. Our quails are at day 9 of incubation, so we're getting closer to our hatch date, which is approximately next Tuesday or Wednesday, I'm thinking. However, like I mentioned last class, our temperature dropped a little bit at the beginning of embryonic development, so we may have slowed down their growth. So we're anticipating signs of hatching maybe next Wednesday or Thursday, but you can see they've grown quite a bit from looking like that weird little tadpole to now resembling something that's similar to a bird with giant eyeballs. All right, let's get to our notes. So we've been talking a lot about combining alleles. If we know the parent genotype or combination of alleles, what could possibly happen during sexual reproduction? And we've been kind of like visualizing, is it going to look dominant? Is it going to look recessive? Let's try to find out, especially with that worksheet with the dogs on the back. Let's combine these two alleles and see what happens. This is going to be so much easier. So a Punnett Square is used to predict the possible gene makeup. Rather than thinking about what it could be, let's prove it using something called a Punnett Square. And this is what it looks like. It's simply a grid where you put one parent genotype along the top, one parent genotype along the side, and then you combine them going across or down. The only thing you have to write on this slide is the red terms that are underlined. So we only wrote down a definition. A Punnett Square is used to predict the possible gene makeup of offspring. We can do these predictions in our head, and many of you can already do that. However, a Punnett Square is used to prove your predictions and show the likelihood of getting different genotypes in offspring. And here's how we set them up. At the top of the screen, you see it says male, X, female. This is how we represent the two genotypes that are used in a test cross, is what we call it. So sometimes it's a plant, sometimes it's an animal. It can be any living thing that is reproducing sexually, because that's what the Punnett Square represents. What could possibly happen during sexual reproduction? If we know the genotype of the male, and we know the two alleles for whatever trait we're looking at, we write them along the top. And this represents the possible allele in every single sperm cell or pollen. And then, of course, we put the female gametes on the side. And I want you to notice that there are two little red lines along the top, two little red lines along the side, because we actually separate each letter, because you can only pass down one form of the gene during reproduction. So we know that whatever species it is, we're kind of assuming that it's a 2N organism with diploid cells. We're splitting apart the chromosomes because during reproduction, you can only pass down one or the other. And then inside each of those boxes are the new combinations, which lead to genetic diversity. One from the male, one from the female. Here's what the offspring could possibly look like. After we do our test cross and after we fill in the Punnett square is when we work on the genotypic and phenotypic ratios. And we're going to practice this together. It's probably the most difficult part of using a Punnett square is taking the information we see and writing it in the form of a ratio. So a genotypic ratio is a comparison of the three possible genotypes we can get. We know we can have homozygous dominant, 2 capital letters, heterozygous, 1 capital, 1 lowercase, or 2 homozygous recessive, which is 2 lowercase letters. And that's the sequence we're going to write it. So it's a comparison of homozygous dominant to heterozygous compared to homozygous recessive. So that'll show you how many of the offspring will exhibit each genotype. For phenotypic ratio, you're simply comparing the dominant to the recessive. So both forms of the gene. How often do we see that dominant form? How often do we see the recessive form? So we compare it. How many of the 4 are dominant? How many of the 4 express that recessive form? So let's do a practice. If we are looking at mice and we're just kind of creating these scenarios for the purpose of practicing our Punnett squares. In this case, we're looking at fur color. And if we're telling you that black fur is dominant to white fur, we're going to use B to represent this gene, capital B for the dominant allele, lowercase b for the recessive allele. I'm telling you that a male mouse is heterozygous for black fur, and that tells us a few different things. First off, he's got black fur. That's what we see. But it's not enough to set up a Punnett square. You have to know the exact genotype. So if I tell you it's heterozygous, we know that he's got 1 capital B, 1 lowercase b. He's also called a hybrid for black fur. So he does have that recessive there that's hidden. The female is homozygous recessive, and that tells us a lot about her. First off, she's got white fur, which means the dominant allele is not present. 2 lowercase b's. Now, we've talked about this in the past, no matter what class you're in listening to this. If both parents have at least one recessive form of the gene, it means that some of their offspring, we don't know exactly how many, but some of them will definitely express that recessive form. Because sometimes that hybrid or heterozygous parent will give the dominant, but other times they're going to give the recessive as well. So let's see how this plays out in a Punnett square. So what we're going to do is the black fur color is dominant over white fur. So we're using capital B for black, lowercase b for white. The male's genotype is capital B, lowercase b because he's heterozygous. And the female is little b because she's homozygous recessive. We're going to take that male genotype and separate the alleles along the top of our Punnett square. Take the female alleles and separate them along the side of the Punnett square. This means that this capital B will be used in the boxes below it. This lowercase b will be used in the boxes below. This b will be used in the boxes across. And this lowercase b will be used in the boxes across as well. So when we get heterozygous big B, little b, it's because it's a combination of one allele from the male and one allele from the female. These two intersect right here, so we get a heterozygous, and of course we always write the dominant first. So one possible offspring could have black fur if it's a hybrid. Continuing across, this little b is used in this second box with this little b that's used in the boxes below it. So we get a homozygous recessive, and that means we do have a possible offspring with white fur. This capital B is used in the other box below it. with this lowercase b that's used across. So we get another heterozygous black fur offspring, continuing across with that little b and combining it with this little b, and we get another homozygous recessive white furred offspring. So looking at our Punnett square, we see that we don't get any homozygous black fur. It's not possible to create a purebred capital B, because that female only has the recessive. However, when we write our genotypic ratio, we have to remember to include that. So our genotypic ratio is always written in the same sequence. In this case, we get 0 homozygous dominant, we get 2 heterozygous, and we get 2 of the 4 homozygous recessive. For our phenotypic ratio, we're comparing how many black fur to how many white fur. And in this case, there's 2 black fur and 2 white fur. We can see them right here in the Punnett square. It supports our conclusion. If we were to cross these 2 mice, this is what we think would happen. Half of the time, their offspring would be heterozygous with black fur. The other half of the time, they could be homozygous recessive with white fur. It doesn't mean this is exactly going to be true, but this is what we predict would happen most of the time, or our numbers are going to be really, really close to that. When we go to percents, this is where it gets a little bit tricky, but you're only ever working out of 4. So with genotype big B, that's 0% of the time in this case, because we don't see it in the Punnett square. But 2 of the 4, or 50%, are heterozygous, and 2 of the 4, or 50%, are homozygous recessive. That means that 50% of them are black and 50% have white fur. So that's how we use a Punnett square, and that's how we write our percentages and our ratios. Let's do another practice. Let's cross 2 heterozygous mice. Yep, this is the next one. 2 heterozygous mice and give the genotypic and phenotypic ratios. In this case, it doesn't matter which one's male, female, because they both have the same genotype. And if they're both heterozygous, it means that they both have a dominant and a recessive. So we can make some predictions that we know that some of them will probably both get capital letters, some of the offspring will get both capital letters. We can also predict that some of the offspring will probably get 2 lowercase letters. So we're going to have a combination of all different genotypes here. One parent goes along the top, one parent along the side. And remember, this capital B is used all the way down. This lowercase b is used all the way down. This capital B is used across. This lowercase b is used across. So when we combine these 2, in this first box, we have homozygous dominant. When we combine these 2, we get heterozygous, but both of them still have black fur. When we combine these 2 here, we get another heterozygous, so still black fur. But this last little box, we have little b combined with little b from both parents, and we get the reemergence of that recessive form of the trait. So the only way to get a mouse with white fur is if both parents give that form of the gene. For our genotypic ratio, we do have one big B, one homozygous dominant, compared to 2 heterozygous, compared to 1 homozygous recessive. So we get a 1 to 2 to 1 ratio. And for percentages, 25% are homozygous dominant, 50 are heterozygous, and 25% are homozygous recessive. For phenotype, remember, we're just comparing black fur to white fur. We've got 3 black fur to 1 white, or 75% black to 25% white. And this is where I'm going to stop for our video. However, there are a couple more examples. I'd like you to try the next one on your own. We're going to assume that for plant height, we're going to use capital T for tall, dominant, and lowercase t for the recessive form, which is short. So try that next one on your own. Give me the ratios, and then call me over when you're finished, and I'll check it.